So again, being a journalist during the pandemic, I have to confess I fell in love with T cells. They are pretty amazing cells. Writing about infection, disease, and immunity helped me discover the many wonders of these fighters that have memory and can fight diseases even years after finding a pathogen. It is also amazing how science has managed to train these T cells to create CAR T cells against cancer. So the work of our next speaker was key for our knowledge of infection, immunity, vaccine development, among the others, all centered in T cells. Dr. Matt Davis is the director of the Stanford Center for Human Systems Immunology, a professor of microbiology and immunology, and a Howard Hughes Medical Institute investigator. He received a BA from Johns Hopkins University and a PhD from the California Institute of Technology. He later was a postdoctoral fellow and staff fellow at the Laboratory of Immunology at NIH, and then became a faculty member in the Department of Microbiology and Immunology at Stanford University School of Medicine, where he remains today. Dr. Davis is well known for identifying many of the T-cell receptor genes, which are responsible for the ability of these cells to recognize a diverse repertoire of antigens. Please join me in welcoming Professor Davis, who will speak on enabling human immunology.
Well, thank you very much. And it's a great pleasure to be in Madrid again and visiting the CBM again. And I've been a frequent visitor, thanks to Albino. And it's also, of course, an honor to be helping you celebrate the 50th anniversary, which is fantastic, and to be in this fantastic meeting. And hopefully I can control slides and things. Yeah, okay. All right. So not going to talk about T-cell receptors, but I think a bigger problem, which is that modern immunology has been really built on the inbred mouse model. And it's for 60 plus years, this has been the principal way in which we understand immunity. But we know it's not the whole story because we see all differences. We try to model human diseases, and most mouse models of human disease don't work. They don't produce actionable intelligence, much less cures for diseases. Some have been terrific, but by and large, they're not predictive. And what Lewis Carroll might ask, when is a model not a model? And I think when it's not predictive, it's not a model. You need to move on to something that is predictive. And of course, we know the inbred mouse was designed to have no genetic heterogeneity. And that was a reasonable sacrifice to make because some of the other things are so complicated. But we also know that inbreeding is bad for you. And I have to tell people, if you're thinking of inbreeding, don't do it. But there's also, of course, a lack of microbial and disease experience. We keep mice in the basement wrapped in HEPA filters. It's really not I've never seen an immunologist so angry as someone tells them there's a disease in the mouse house, that the mice are coughing. No, please. So we it's an artificial organism in an artificial environment, and it's been a fantastic tool, but it's just one tool. And maybe we should move on to a bigger, more complex organism like humans. And also we know, I think the biggest thing is that there are huge differences in longevity and fecundity between mice and humans, and all suggesting, this is not working, that the human immune system is going to be quite complex, much more complex. And we also know the humans did something spectacular, talking about the evolution of humans. They did the worst possible thing for infectious diseases, which is to invent agriculture and to live in very dense communities that are ideal for the spread of infectious diseases, and trading between population centers, so that a plague in thousands of miles away could come to your doorstep. and infect you. And so, and that was why I think that almost half of children under 5 died of infectious diseases back before childhood vaccines and so forth. So, I mean, we really put ourselves under tremendous immunological pressure. And that was in the non-plague years. When the plagues came, that was actually got even worse. So anyway, I think based on the failure of mouse models of disease and based on obvious differences that you need to have a more capable and complex immune system that can defend you against infectious diseases for years and years, that this seems a no-brainer. Plus, we've also seen a lot of failures in vaccines. The COVID vaccine, of course, has been spectacularly successful, but there are many failures in the more complex diseases like TB and malaria, dengue, HIV, and we developed drugs, the vaccines based originally on small animal models that you don't really test vaccine variants in humans or even primates. It's just too difficult. And so we I think really desperately need a more accurate models of vaccine development. I'd also want to spend some minutes on influenza. Influenza is a really crappy vaccine that's never better than 60% efficacy and often worse than that, but we'll talk about that in a minute. So what to do if you want to understand human immunology? Well, one is there were almost no tools 20 years ago. And so we spent a lot of time developing new tools, informatic tools, physical tools, better, more complex staining of single cells. Single cell methodology has increased tremendously. The technology we have now to look at the immune response at a single cell level is really fantastic and light years away from what we were. And I've also managed to persuade a lot of colleagues at Stanford to also help us develop these tools. But the recent tool that I think is the most powerful of all has been that we found a few years ago, that we can take human tonsils, which are routinely excised from both young children and adults for sleep apnea, have a huge number of lymphocytes. And so you can make a suspension of single cells from tonsils and reproduce in a dish vaccine responses, at least some vaccine responses, and get germinal centers and antibodies and T cells specific for, in this case, a flu antigen. We've seen evidence of somatic hypermutation and selection of high affinity antibodies. So it gives us a way to look at human immunology in a dish, and it's in vitro. So it's even better. Typically, in vitro is much more powerful than in vivo. So even a mouse isn't as powerful as a system, much less that we know we're looking for human specific differences. So what have we done with this system? And some things have worked out really well. And particularly the problem of flu vaccination is one that's not talked about much. But the reason the flu efficacy of vaccine is so poor, it's not, the other vaccines are 90, 99%. And so having 60% efficacy is really terrible. And this has been the situation for decades and decades now. And why hasn't this problem been solved? Well, it all goes back to the dominant model for why flu vaccines are not efficacious is the phenomena that most people have a strain preference. And so even you get a vaccine that has 3 or 4 flu strains in it, you typically will only make a vigorous response against one of those strains. And this was a big mystery. And then this guy, Thomas Francis, one of the pioneers of flu vaccination, had an insight that he called the doctrine of original antigenic sin. And I think part of the lesson here is if you want to make a model popular, introduce some aspect of naughtiness that, and so this has been the dogma for 60 years to say the reason we have a strain preference is because the first flu infection you ever got biased your immune system towards that strain. And so ever after, you're going to prefer that strain. And it just turns out this is not true. And this guy, a postdoc in my lab, Vamsi Malajosa, figured this out, and he did a number of population studies, twin pairs, and unexposed infants. Basically, showed this isn't really true. There is memory. If you get a flu infection, the next year you will have a preference for that infection, but it's not the main driver of strain preference. So what is the main driver? The main driver goes back to some basic immunology where how do T cells and B cells interact with each other? And one model that's quite true is that a B cell with a particular specificity for, say, a flu strain will grab hold of that antigen, pull it in, break it into peptides, and those peptides are presented to on MHC, bound to MHC molecules that are then seen by the T cell receptor on particular T cells. And this interaction is critical for whether you get good antibodies or not, because it's without the T cell help, as it's called. The B cell typically cannot mature, cannot secrete antibodies, cannot mutate the antibodies to produce higher affinity versions. So Vamsi figured out that this might be due to MHC class II polymorphism. And he went, there's a great resource called the Immune Epitope Database, where you can basically plug in any amino acid sequence and ask how many peptides will a given MHC process and present from that is capture and bind efficiently based on a sequence motifs and things like that. It turns out it's very heterogeneous. That you can see that the white is more peptides, the dark is less peptides. So you could see different class II MHCs, and there's huge polymorphism in a population, have different preferences. And he confirmed this was likely to be true based on some peptide experiments, but what was really convincing was to do an experiment with tonsil organoids and basically give the tonsils two different versions of a flu vaccine. In the lower case is the standard flu vaccine where you grow up different strains and then you put them together. So they're independent collections of the antigen for that strain. But the variant he made was he put all of the antigens, the different antigens, on one vector. And that worked. And so if you look on the left side, the IIV is a standard flu vaccine with 4 strains that most of us will get every season. And what you can see is that with this particular tonsil, there was the H3 strain is by far favored based on antibody response, and the other strains are not well represented. When we did this mock-up with the tonsils, with the same tonsil, we basically saw the same thing when we had separate vectors with the different strains on them. But then when we combined all the strains, we actually got equalization of the response. So we cured the tonsil of strain bias, basically. Why is that true? Basically, what we're doing is the B cell is absorbing, if it only absorbs one strain, it'll only present peptides to T cells and get T cell help from that strain. If that's a popular strain in that person, that's fine. But if it's an unpopular strain, it has very few B cells and very few T cell help. But if you combine all the strains in one vector, then every B cell that has any flu specificity can pull in all of the strains, proteins, and make peptides for all of those strains. And so even its a least favored B cell, it'll get T cell help from the most popular strain. And that's the key, because T cell help is limited. But somehow I am talking about T cells. Yeah, I keep going back. I don't know why. So, just to say how useful this could be in terms of development, bird flu, avian flu is probably, has been a candidate for the next pandemic, in case you've been keeping track. And there was a pandemic maybe a few years ago, you remember? So the bird flu can be, this could be a devastating pandemic. It has a 50% mortality in the Asian countries. It's the one that has gotten to the North America anyway is much less potent, but still, it still would be a big deal and at least as bad as COVID. So again, if you see, it's a little hard to see, but if you just take avian flu hemagglutinin, which is the most immunogenic part, and you immunize tonsils, you get crappy response. Even if all the tonsils have seen lots of flu, they haven't seen the avian flu. And so the response is, in terms of antibodies, is very bad. But if you make a vaccine construct that targets avian flu specific B cells, which will be very few, but brings along the more popular antigens from the other flu strains, now you get T cell help, and now you have a substantial, a little purple bar there, and on the far right is example. So now you can get a substantial amount. Basically, you're borrowing T cell help from the popular strains to boost up the antibody response. So this is, I think, really will help get a flu vaccine into something useful. Another area that we've been using the organoid system effectively is to chase after a phenomena we saw years ago now, where we saw that in both celiac disease and in a mouse model of MS, we saw a kind of funny reaction or collection of alpha beta, CD4, CD8, and gamma delta T cells coming up in celiac disease or in this mouse model. And long story short is that basically there are these a whole other type of regulatory T cell, which is of a CD8 flavor, and it's cure positive in humans, live 49 positive in mice, and it's a big deal in autoimmunity. We can see normally it's only 1 or 2% of the CD8 cells, but we can see at very high levels in, say, lupus or MS or celiac disease in some patients. And Jing Li has been, was a fantastic postdoc working on this. She now has an independent position in Pittsburgh. And basically, we also could see that this was these regulatory T cells, which can kill celiac-specific T cells, for example, are also elevated in infection, whereas the more classical regulatory T cell, which is of the CD4 type, been around for 30 years, very important in terms of tolerance, but it's not active in infection. The CD8 regulatory cells are very active in infection, probably because infection affects your barrier for tolerance. You're allowing self-specific cells to become active where normally you don't. But you do this in an infection because you might die and you might need these cells for to defend yourself. We don't need to know that. So we had to go back. This is one of the tragedies of human immunology, is that if we find something important and interesting in humans, we almost have to go back to mice to show referees that it's really important. So sort of it's an animal sacrifice that we have to make in order to sanctify human data. But long story short is that we knocked this out in mice, these regulatory CD8 cells, and they develop autoimmunity after an infection. Okay, so they're also active in pregnancy. So in this data, we show that women that are pregnant with a male fetus have elevated levels of this CD8 regulatory T cells. So it's active in fetal tolerance, very pronounced. And then in preeclampsia, we're also seeing elevated cells. So it seems like if we have a problem, especially the one that might be autoimmune, you're going to see an increase in these cells. So it's sort of a bioindicator of a autoimmunity problem. And so we're really interested in using the tonsil system and other systems to understand the difference between the CD4 regulatory cells and these CD8 regulatory cells. Again, we're just starting to understand that it's a very new phenomena. And so a brave grad student, Mustafa Banazada from Denmark and Xin Chen, a postdoc in the lab, developed a knockout protocol using CRISPR-Cas9. And basically, if you knock out FOXP3, which is the main driver of tolerance in the CD4 regulatory cells, now the tonsils will make autoantibodies, which they wouldn't before. Normally, they're suppressing. They have the B cells to do this, but they're normally suppressed by the regulatory CD4 cells. you knock that out, and now you get autoantibodies. And so we basically can create a model of autoimmunity in the entirely human tissue, immune cells. If we knock out the CD8 regulatory cells, we don't have an effect on the antibody production, but now we see that autoreactive CD4 cells are allowed to proliferate and become an item. And if you combine it with the other data, you could think of a two-step model for autoimmunity, which is we know that in most cases of autoimmunity, you see autoantibodies years before clinical autoimmunity. But what you see just before clinical autoimmunity is infection. And so we think you're first have to get past the CD4 regulatory T cells to make autoantibodies, but then later with certain infections and maybe other circumstances, you can go the final distance, break CD8 T cell tolerance, and there you have a disease. So, and then lastly, we've been really struck by collecting other immune organs, and it turns out that it's very easy to get human spleens from transplants. So when surgeons go to harvest organs from a donor on life support, they take the spleen along with everything else. But no one wants to transplant spleens. It would be a really bad idea, actually, to give somebody a spleen that doesn't belong in your immune system. And so they throw it away. And yet these are huge. You get 10 billion cells. And so we can freeze down dozens and dozens of aliquots, and also they're more diverse and give better vaccine responses. We can see history. We can see measles-specific B cells in spleens from people that are 50, 60, 70 years old. So this is persistent, the memory of immunity. And so we've been developing the spleen bank of Palo Alto, and we have over 100 spleens now that we have banked, and it represents the human immune diversity. We know humans have very different immune systems, and that's the evidence is if you give an immune drug to anybody, they may respond, but someone else won't respond. And there's a huge heterogeneity here. It's a huge medical problem to know what drug to give to what person. And so we're very interested in looking at the human diversity using this spleen collection, and things are coming out that are, I think, going to be very important and useful. But lastly, what we're also trying to do, and why organ donors are a key part of this, is that we can get other human tissues from the same donor. And you need to have the same donor to go with the immunology. And so we've been making not just spleen organoids from donors, but lung organoids from the same donor. And this has been technology developed by Calvin Poe at Stanford, who's a fantastic organoid guy. And so he's developed a very nice lung model, which contains a lot of immune cells. And my student Azim has been culturing spleen and lung organoids from the same donor, and then infecting the lung with a pathogen and looking for an immune response. And so this is some of that early data where you can, if you infect the lung, you co-culture the lung organoid with the spleen organoid from the same donor. If you infect the lung with flu, you can trigger an immune response in the spleen, both antibodies and T cells. So really getting a model of infection and of communication between an immune organ and a tissue that has some problem, in this case, infection, but it could be cancer, it could be autoimmunity. So in conclusion, I think, if I'm on time, it's a miracle. But anyway, I think a major problem in human immunology has been that we haven't been able to do mechanistic experiments in a human organ or in a human context. And you can't do things with living humans, that there's laws and so forth that prevent you from doing very much. But with the organoids, you can do everything. And you can do everything at a single cell level and look at things in tremendous depth, but also being able to knock out genes, to manipulate the cells, to add cytokines, to take away cytokines. It's just a perfect system for getting into depth and things, and finding human-specific immune mechanisms and responses and heterogeneity that there's no way we're going to be able to do with mice. So I think these systems are not the answer to everything, but they could be. It can give us a huge start on trying to identify and understand things. I mentioned the problem with blue strain preferences and the connection with T cell help in MHC class 2 polymorphism. And, I think in this case, religious institutions have been trying for thousands of years to abolish sin without obvious success. But here in the vaccine case, we can remove sin from the equation. I think I'm very, very proud of this and anxious to tell people. Anyway, and then the problem of T cell regulation, as I mentioned, we're able to address this in an entirely human system. And actually there's a paper in Nature Immunology on this. It's the first paper we published on human immunology where they didn't ask us, they didn't demand to do a mouse sacrifice. So I think we really crossed the barrier and very proud. So anyway, and then lastly, I think combining immune organs and those and looking at that communication between an immune organ like a spleen and a tissue that's having some issue and is communicating and getting help from the immune tissue. I think that's going to be important, and also to develop vaccines. It's you can do this. You could do 1,000 assays. You can prototype vaccines, in a human system that we can never do previously. So I think it's going to revolutionize vaccine development and get us maybe effective vaccines to some of these more difficult diseases. And thanks, I have fantastic students and postdocs and technicians in the lab. I mentioned Vamsi, I mentioned Shin, Chen, Mustafa. Others have been tremendous, Azim. But I have a really great crew of people, great collaborators like Calvin Kuo and Chris Garcia, Bill Robinson, Christine Jacobs-Wagner, others, and also patient funders like the Gates Foundation, NIH, Open Philanthropy, and of course our donor network West is the provider of the spleens and other tissues, and has been a great collaboration there as well. So I'll stop and answer the questions.
Thank you, Omar. A couple of questions which I think are related. What's your opinion about microbiome in all this vaccination, and especially related to the fact that in some studies, non-inbred mice are better model for immunology responses?
Yeah, I mean, I think I understand the question. Definitely more genetic heterogeneity, getting away from this purely inbred model that's very constrained, like the Black 6 model, which you can't do this original sin experiment on because it has one MHC class 2. So, I mean, it's designed not to give you the right answer. But yeah, you can definitely, and people have crossed mice, and there's Swiss outbred mice, totally not popular because you can't use any knockouts with them. So partly a lot of modern mouse immunology is built on this huge collection of gene knockouts that you can access and bring in. And so that is so attractive to people. They don't use those other models. And the other thing is there's still mice, and mice, if a mouse lives for 6 months, that's fine. You're going to have a lot of mice. If you can't get a human to survive for 20 or 30 years, you're in big trouble as a species. So I think in terms of the next 100 years of immunology, it'll be human, and it'll be about uniquely human mechanisms and science. So I think it's actually a perfect sort of marriage of basic science and the drive to discover things with translation, because we need better vaccines. We need to understand human cancer and the immunology of human cancer. We don't, we barely scratch the surface of this, partly because we've been so dependent on mouse models. But now if we can get away from that, get into looking at actual human responses and human memories and diversity, I think we'll be in a whole other area. So
Mark,
oh yeah, Elaine.
yeah, I really enjoyed your talk. The only comment I might make is that from someone who spent the first 10 years of her assistant professorship working on organoid cultures and switching then to mouse, is that for a tissue, for an organ like a lung or the skin, which you'll hear about this afternoon, there are 50 different cell types, as we just heard, also coupled with the microbiome. And so I'm hoping that in your organoid collections, you are saving all of those other 50 cell types, because you may need them if we're going to understand immunology.
No, absolutely. I mean, as I said, that was there was I protected myself by saying this may not be the answer to everything, but it's a beginning. And then then you add the microbiome influence, you add a nervous system influence if you can, and there are going to be limits. I think every system has some limits and some compromises, and you should only work on the things that you can validate are actually happening in a whole organism. But you got to start somewhere, and the resolution that you can get in a dish is, especially with the modern technologies, is so powerful.
The only thing, I think, if you're banking all those other cell types, then it would actually really guide you as you start to realize you're going to need this or that. You'll have it.
And that's also the beauty of biology, is you don't really know what you're doing. You're just kind of zigging and zagging and say, oh, well, okay, all right, I didn't know that was there, and then we'll study that. So I think it's not a linear profession. It's well, well, also I have to explain, we talked about molecular biology yesterday. I had to once explain to Tommy Aniatis, who was on my thesis committee, that whereas molecular biology came from physics and chemistry, immunology comes from witchcraft and shamanism, where the important thing is to know the secret name of something, and then you gain power over it, and at least your colleagues, perhaps.
So great talk. And along the lines of witchcraftery and your abolishment of the sin, congratulations to that, because breaking dogma, it's a hard thing to do, right? But I was curious about when you created those linked flu, I guess, vaccine prototypes.
Yes.
Does it matter which linkers, how many, what order?
No, we didn't test that. I mean, the main contrast was between a heterogeneous vector with all the different strain type antigens with what everyone does. Every multi-strain vaccine, including I think pneumonia has like 19 strains. They're all made the same way. They're all grown up separately, and then you mix them together. It's like wine. You kind of mix the different strains together and hope that's going to work. But it doesn't work because of this MHC polymorphism. So I think it's partly, I think the the science of vaccines really needs work, and people did what Pasteur did, and it worked, and there's a lot of success that came from how it was done early on. But I think we're now dealing with much more complex organisms, and I can tell you about TB and malaria, for example, where we can show that actually most of the T cell responses are irrelevant to protection. And so that strikes at another dogma of vaccinology, which is you choose the most immunogenic antigens, and you need immunogenicity. You need a strong immune response. But it turns out a lot of these antigens that the TB organism and malaria parasite are throwing out are distractions. I call them distractopes, actually. They're what the pathogen wants you to make a response to keep your immune system busy and hiding some of the more important antigens that might be much more effective in vaccines.
Interesting you say that, because one of our approaches was first we looked at what these hookworms and larvae were shooting out.
Yeah.
Total failure. We had to go into what we call the hidden antigens, and
Yeah.
we magically.
No, exactly. This thing, yeah.
And so your prediction is mosaic
Yes.
like
Yes.
production system, which is very complicated to produce and
Yeah.
scale
Yeah.
and-
No. It's a job. Yeah.